Welcome to Study Time, a televised home learning program produced by Rwanda Education Board.
Hello students, welcome to the TV learning programs. As usual, I'm teacher Hurbert, who is going to be taking you through Biology Senior 2, which is concerning about enzymes. Biology and health sciences. Senior 2, and write it here, Senior 2, which is Unit 7, concerning about enzymes. So students, let's have, before we start, what are enzymes? I know some of you can have some stains on your clothes. When you have a stain on your piece of your clothes, sometimes it is hard to be removed when you don't have a detergent. But when you have a detergent, it is easy to be removed from your clothes. So enzymes also act as detergents, but what they normally do, they speed up the rate of chemical reaction, and they don't initiate the reaction to start. So before we are going to start that lesson, I have to remind you to have a pen, an exercise book, and a notebook. And for those who have the textbooks, you can check on page 84 on what you call Biology and Health Science for Rwanda schools. So the learning objectives of that lesson of today, these are the learning objectives. Learning objectives. Number one is to understand what are enzymes. That is one learning objective, number one. Number two, what are the importances of enzymes? Objective 3, you have to talk about properties, can call it be properties or stroke characteristics of enzymes. Okay, then let's start the lesson. What are enzymes, students? What do you understand by enzymes? I said that enzymes are related to the detergents because I said once you apply a detergent to some of the clothes that has a stain, the stain has to be removed by the detergent. So it means that enzymes are also able to speed up the reaction. So enzymes, by definition, you can say enzymes are biological catalysts, which are protein in nature. If I say protein in nature, specifically I mean they are globular proteins, and they can speed up the rate of chemical reactions or they slow down the rate of reactions, but they remain unchanged at the end of reaction. Okay, you can say they remain unchanged at the end of the reaction. So here is the definition. You can say enzymes. The definition of enzymes, you can say enzymes are biological catalysts. If I say catalysts that are globular proteins, if I say globular proteins, we have two types of proteins. We have globular proteins and fibrous proteins. Globular proteins, most of the time, they normally participate in metabolic reactions. And then for the case of the fibrous proteins, they're responsible for the growth of hair and nails. All right? And I can say protein in nature, and protein in nature, and speeds up or slow down the rate of chemical reaction. Okay? Chemical reaction. So slow down the rate of chemical reactions in the body without being used up, without being used up themselves. Simply, they remain unchanged. So that is the definition. You can take a note about the definition of what you call enzymes. All right? I said enzymes are biological catalysts that are globular protein in nature, or they are protein in nature, and they speed up or they slow down the rate of chemical reactions in the body but without being used up themselves, or simply they remain unchanged. So there are two types of enzymes in our body. Okay? You can say types of enzymes. Types of enzymes. Types of enzymes. We have two major types of enzymes. One are intracellular enzymes. Number two, we have extracellular enzymes. These are enzymes, this one also all enzymes. So let me start with the intracellular enzymes. Most of the enzymes are normally produced by the cells. And once they are produced by the cells, they have to work inside the cell, or they can work outside of the cell. So this type of enzyme intracellular enzymes are produced inside the cell, and work inside the cell. Example, we can have example of intracellular enzymes. We have these are alimentary canal enzymes, which can be produced in alimentary canal, and they play a big role in the digestion of food. We also have another one. We have what you call respiratory enzymes. Respiratory enzyme. We have examples of. Let me just have examples of intracellular enzymes. Examples of intracellular enzymes. We have respiratory enzymes. Enzymes that are more produced in the respiratory system. We also have another one. We have what you call the catalase. The catalase, which is commonly found in plants, and in liver of animals. So these enzymes are produced inside, but they have to work outside. So we have number two. This was number one. Number two, we have extracellular enzymes. These extracellular enzymes are produced inside. Extracellular enzymes. These are enzymes that are produced inside the cell, but are secreted outside of the cell. All right? Let's have the examples of extracellular enzymes. So I think you can take a note about that. You say they are produced inside, and they work inside. Example, I talked about respiratory enzymes. I talked about catalase in plants, and even in the liver. All right? So then extracellular enzymes, they are produced inside the cell, but they are produced or they are secreted outside of the cell to perform its function. All right, let us have the examples of extracellular enzymes. Examples of extracellular enzymes, examples of extracellular enzymes, extracellular enzymes. Enzymes we have one, we have what you call elementary canal enzymes, which performs the role of digestion of food. Digestion of food, because we know that we have two types of digestion. We have chemical digestion and mechanical digestion. So for the case of chemical digestion, it's normally done by the enzymes that are found in the alimentary canal. We have the salivary amylase, we have the pepsin and renin, which are found in the stomach. So we can also say that another example, we can talk about another enzyme which are normally produced. We have what we call retic enzymes. These retic enzymes are produced by the fungi and bacteria. They are secreted inside, they are extracellular, they are secreted inside the cell, but they go to work outside where they normally help in the process of decomposition. So most of the decomposers, we have the bacteria and fungi, and for them to participate in the action of decomposing some organic materials are normally facilitated by retic enzymes, which normally breaks down dead organic matter into simple soluble compounds which are going to be used by the cells. We also say that there are also some of the enzymes that are found in living tissues. In living tissues, examples of living tissues, we have the catalase enzyme, which is found commonly in the liver, all right, which helps in the breakdown of hydrogen peroxide into water and oxygen. Here we can have the equation. This is the equation of the reaction. You get hydrogen peroxide, and you add an enzyme which is called catalase, which is going to be able to speed up the rate of chemical reaction. So you add the catalase enzyme, and there you have to get water, a liquid, then plus oxygen gas. So as you increase the catalase there, it has to speed up the rate of chemical reaction in the liver, and also these catalase can also be found in Irish potato. Irish potato, we can also extract their catalase. Quickly, let's talk about other types of enzymes that are found in our body, and they perform a function of breaking down food into simple particles to be absorbed into the body, and they normally named according to the food they act upon. These enzymes, we have a number of other enzymes that we are going to mention. Other enzymes that we're going to mention. We have one which is called protease, the protease enzyme, which breaks down proteins. Students, I know you know what are proteins, breaks down proteins into peptides stroke amino acids. I know the product of proteins are amino acids, because the amino acids are the final product of proteins. Number two, we have another form of enzyme which acts on lipids, which we call lipase. And before I talk about protease and lipase, most of the enzymes, they have a suffix which is called ase. The presence of ase indicates it is an enzyme. When you get anything and you add something like ase, catalase, protease, lipase, it means it is an enzyme. All right. So lipase, it breaks down lipids into fatty acids and glycerol. Good, fatty acids and glycerol. All right. We have number three, which is called carbohydrates. Carbohydrates. These are enzymes that normally act on carbohydrates. which breaks down carbohydrates into reducing sugars. Take an example. They convert starch into maltose. All right? So we also have other enzymes that are found in our body. We have some enzymes that are found in the stomach, but for them they don't have a suffix which is what it is. We have renin, we have pepsin, which is found in our gastric gland. We have also the renin, which is also found in our gastric gland. The pepsin acts on protein. These are the pepsin and renin. Both of them they are referred to as protein digestive enzymes. Good. Let's talk about now characteristics of enzymes. Quickly, let's look at the characteristics of enzymes. There are a number of characteristics that indicates enzymes. We have a number of characteristics or number of properties of enzymes. Let's say, take a note, characteristics stroke or properties of enzymes. All right? So I can say one of the properties of enzymes, we can say enzyme number one, the enzyme they are protein in nature. Number one, enzymes are protein in nature. If I say they are protein in nature, it means that all enzymes are protein in nature. And it means that the enzymes are made up of proteins. Number two, we can say enzymes are affected by temperature. Enzymes are affected by temperature. So it means that enzyme must be having a particular temperature that has to perform its function. If I'm trying to draw, I can draw it like that. This is supposed to be the temperature. And we can say the temperature could just be going from 1, 2, 3, 4, maybe up to 40. And then I have this supposed to be the rate of reaction. So as the temperature increases, also the rate of enzyme catalyzed reaction increases. It increases as the temperature increases. But when it comes to exceeds to optimum temperature, the optimum temperature must be ranging to 40. All right? When it comes to here, that's supposed to be maybe 40 is here. All right? Let me say this is supposed to be 10, this is 20, this is 30, this is 30 up to 40. When it comes to here, the enzyme catalyzed reaction are affected. Let's say here you can say the enzymes are denatured. But also when the temperature is very low, the enzymes becomes inactive. So high temperature denatures enzymes. Why do we say that it denatures enzymes? The enzymes are protein in nature. High temperature denatures enzymes. That's why the body does by all means to do what we call homeostasis, to balance the body temperature, regardless of external factors. However much the external factors is too hot, the body has to maintain the body temperature so that it cannot exceed 37 degrees Celsius. If it exceeds 37 degrees Celsius, the enzymes are going to be affected in the body. And again, when the temperature is very low in our body, the enzymes are going to be inactive. Let's have another function. We say enzymes work at a specific pH. Another characteristic, we may say enzymes, that is supposed to be number 2, that is supposed to be 3. Enzymes work at a specific pH. If I say a specific pH, automatically if I say pH, it refers to the degree of acidity and alkalinity. So most of the enzymes, they normally work to different mediums. The different mediums I'm talking about is this. I'm talking about a neutral medium, I mean acidic medium, and also talk about acidic medium. most of the protein digestive enzymes, they work their functions in acidic medium. All right? And these carbohydrate enzymes, they normally work in a neutral environment or in alkaline environment. All right? Let me say this is the graph which is trying to show you what happens automatically. Then it's supposed to be this is supposed to be pepsin. It is ranging from 1 to 3. Meaning that it performs its function efficiently when the temperature is ranging between 1 and 2. But when it's supposed to be taken an example like pepsin. But if I have an enzyme, I can take an example like amylase, which can perform its function from this supposed to be maybe 4, 5, 6, 7, 8, and above. You may find this enzyme, take an example like amylase, can perform its function in a pH, when the pH is greater than 7. If I say greater than 7, it means that the environment performs its function efficiently when the environment is alkaline. But in alkaline. Then when this one, it performs its function efficiently when the medium where it is acidic. All right. Let's have another characteristics of enzymes. We can say another characteristics of enzymes. You can say enzymes are substrate specific. For enzymes are substrate specific. If I say substrate specific, what does it mean by substrate specific? So it means the enzyme and the substrates must be able to be combining together to form an enzyme complex, an enzyme substrate complex. So enzyme and the substrate must be fitting to one another as a key fits in the door. So enzyme and substrate, let me say this is supposed to be an enzyme, and all enzyme must be having what you call active site, where you call it to be active site, where a substrate will come and bind. This is an example of a substrate, and this is called a substrate. Then from there, after combining, we are going to have a diagram like that. All right, we're going to be having a diagram like that, and we have now. This is the diagram. That one is called enzyme substrate complex. Then after, we're going to be having our enzyme, which is coming back. And I said enzymes, they are used over and over again, and they don't change. Then after, we'll be having what we call, these are called products. Okay? So, another characteristics of enzymes, as I mentioned, that's supposed to be enzymes, they are reversible. That is number 5. Enzymes are reversible. If I say they carry out reversible reaction, an example, let's have a reversible reaction. Enzymes are reversible. That is supposed to be number 5. Enzymes are reversible. If I say they carry reversible reaction. If I get A plus B, I will get, these are reversible arrows, I will get C. C can be converted into A, and A plus B, as well as B plus B can just produce C. So enzymes are reversible. That means that they can change a substance into product and product back into the original substrate. So let's talk about another characteristics. I talk about what you call enzymes. They normally work rapidly. Enzymes, they work rapidly. Take that note. Another characteristics of enzymes, they work rapidly. If it means they work rapidly, it means that they make the reaction to be very fast. Enzymes, this is supposed to be number 6. Enzymes work rapidly. It means that it can speed up the reaction very fast. But speeding up the rate of reaction, that's supposed to be done by the enzymes. That's why I am saying that enzymes, they work rapidly. Enzymes work very fast in converting the substrate into products. And as well as the faster known enzyme is called catalase. I gave an example of catalase, which converts hydrogen peroxide to water and oxygen. So they convert substrate into a product very fast. The last one is enzymes are efficient. Number 7, enzymes are efficient. If I say efficient, what does it mean? I will just combine one word. Enzymes are efficient. It means most of the time enzyme must be used in a small amount. If I say in small amount, it means that it will get more substrate to combine with and form what you call enzyme substrate complex. So meaning that enzyme must be used in a small amount in order to get substrate to combine with. Another one is supposed to be enzymes, they are not used up in the reaction, and therefore they can just be used repeatedly. So that is to say, enzymes are used over and over again. You can say enzymes are used over and over again. And I also want to remind you the last one. We can also say that there's what you call 2-10 rule. 2-10 rule. As you increase the temperature, as you double the temperature by 10, the rate of enzyme catalyzed reaction increases. As you're increasing the temperature by 10, you double the temperature, the rate of reaction increases. That one is referred as 2-10 rule. 2-10 rule, that's supposed to be increasing the temperature by 10, the rate of reaction of enzyme doubles. So students, here we've been discussing about enzymes. We saw the properties of enzymes. We saw the characteristics of enzymes. And we saw what does enzyme do in our body. So I'm going to leave you today with a home assignment which is concerning about enzymes. Number 1 is asking you to distinguish between enzymes and other catalysts. Okay? We also have number 2, which is explaining the meaning of the following terms with the reference to enzymes. Denaturation. Number B is an inactivation. Number 3, the reaction below illustrates an enzyme catalyzed reaction. We have A plus B, and the enzyme X, we've got C. States 2 characteristics of enzymes illustrated in the reaction. Number 4, which is the last number, is trying to ask you to explain why enzymes are said to be substrate specific. So students, thank you very much for being with me in my lesson of today. I will be with you in the next lesson. Have a good day and goodbye.